Good afternoon, everyone, and welcome to today's session of the Expert Reviews in Molecular Medicine Auto Webinar Series. Expert Reviews in Molecular Medicine is an online-only journal featuring authoritative and timely reviews on all aspects of molecular medicine. Our new editor-in-chief, Nicola Curtin, and her editorial team at ERM are keen to promote our authors' articles and these webinars, initiated and organized by two of our editors, Matt Martin and Michael Hewitt. Just for your information, this webinar is being recorded. In a moment, I'll hand over to Matt, who will be introducing today's session with Dr. Paola Dama, our guest speaker. Before we get started, I'd like to go over a few items so you know how to participate in today's event. You have all joined in listen-only mode, which means you will be able to hear us, but not able to speak to us. However, you may send in your questions at any time during the webinar via the Q&A tab, and we will address them at the end of the webinar. Should you experience any technical difficulties, please add this to the questions box, and a member of the organizing team will endeavor to help you. You will also receive a survey after the webinar to know how useful you found it, and another opportunity to ask for any questions. So to kick things off, I'd like to invite Matt, one of the editors at Expert Reviews in Molecular Medicine, to introduce our guest speaker today. Over to you, Matt.
Thanks, Isabella. Welcome, everybody. Our guest speaker today is Dr. Paola Dama, who is currently a research fellow at the University of Sussex in Brighton. So Paola's research investigates the role of coding and non-coding alterations on cancer pathogenesis and progression, with emphasis on development of a new method to identify miRNA targets in pancreatic cancer, guided by CRISPR-Cas9 and next-generation sequencing technology. So before Paola arrived on these shores, she received her Ph.D. in Molecular Oncology at the University of Ferrara, Italy, and the Ohio State University in Columbus in the US. She then commenced her post-doctoral training at the Comprehensive Cancer Center at the, I should just say Buckeye State University, and the University of Chicago Medical Center in the US as well. Paola is also an avid popular science writer and an editorial board member of Cancer Gene Therapies and Frontiers in Transplantation. So just a note, as Isabella said, if you wish to ask any questions, could you please pop them in the Q&A box below, and Paola will strive to answer these questions at the end. So without further ado, could I hand over to Paola, and we're all very excited and looking forward to hearing her talk on a recent publication at ERMM. So thank you.
Hi, thank you for this invitation, and I'm happy to present for this webinar the article that I write in collaboration with the Mohsen Sheikh Hassan that is at the University of Medical Science in Hamadan, Iran. So I can share the screen. Okay, can you see the screen?
Yes, thank you.
Okay. Okay, so the paper that we published in Expert Reviews in Molecular Medicine is DLX6-AS1 as a putative long non-coding RNA candidate in multiple human cancers. So here there is the RNA family diagram where I show the intracellular RNA, and so as you can see that the non-coding RNA is represented the 98% of the entire transcriptome. So, and non-coding RNAs can be categorized into housekeeping and regulatory RNAs based on their function. And most of RNAs except the regulatory function. So in pink, we have that the location is just in nucleus, but as you can see in purple, is also we can find in nucleus and also cytoplasm. According to molecular size, the non-coding RNA can also divide in a small non-coding RNA and a long non-coding RNA. This is what we are talking today. So long non-coding RNA have important roles in regulating the expression of genes. And the dysregulation of long-coding RNA occurs in various pathological processes, including tumorigenesis and also cancer progression. So what is interesting in the long-coding RNA and the mounting evidence that we have in the last years is the fact that can be considered as biomarkers in initial development or different cancer. So here is grouped the function, the mechanism of the long non-coding RNA. As you can see, they can act like guide. So long-coding RNA can guide transcription factors to specific genomic location for the regulation of gene expression, so can suppress or activate the gene expression, and also can act as scaffold to facilitate the assembling of chromatin remodeling complexes. Or as decoy, can also serve as a sponge to titrate microRNAs out from the target and also the transcription factor or other proteins. So there are a lot of function that is reported, and also is all the studies still in development. So we are still learning from the long non-coding RNA and the function. So as you can see, basically cancer is a genetic disease, so in which genetic changes lead to aberrant gene expression. So long non-coding play critical roles in several cancer-related cellular biological processes, such as the cell proliferation, apoptosis, migration, invasion, and tumorigenesis. And also the long non-coding RNA have been shown to be expressed aberrantly in several cancers and play key regulatory roles in cancer. And as is depicted here in this cartoon, we have that they are involved in DNA damage, angiogenesis, metastasis, cell stemness, immune escape, and therapeutic resistance and metabolic disorders. As you can see here, some is represented some of the main known, like MALAT1, NIT1, that is long non-coding RNA. So identification and the characterization of long non-coding RNA in the role of initiate and cancer progression is extremely beneficial for cancer diagnosis and therapy, because the clinical importance of long non-coding RNA has been demonstrated in several studies. Also because a long non-coding RNA can be easily detected from the body fluid, and they can use as the biomarker for cancer. And also, as I told you, is they are dysregulated, so they can overexpress or downregulated. And also can be used now with new technology like a CRISPR-Cas9 approach, which also is a good approach for therapeutics. So we want to discuss about these distal-less homeobox 6 antisense RNA1 that is the family of DLX6. This is why we decided to write this article, because it's one of long non-coding that is expressed is reported in several cancers, as I showed you in a moment. So here, the location of this gene is in region 7q21.3 of the chromosome 6, and also with the gene expression in a genotype tissue expression GTEx database is reported in 54 tissues. So in our articles, so we describe the effect of the non-coding, the DLX6-AS1 in digestive system cancers, respiratory, reproductive, urinary, and the central nervous system, and also other cancers. So is spread is upregulated in most of the cancer. And so we can start, for example, in esophageal squamous cell carcinomas, that is a fatal malignancy. And despite the treatment, the prognosis, unfortunately, is still poor. So we reported 3 papers where they compare also the expression in the clinical specimen, but also in cells. As you can see here from the table, we reported the expression that is upregulated. Also, the functional role of each of our study and the approach that they use, and the target, so the downstream target. So what is interesting in here is there is that one of the role that can have the long non-coding RNA, that they have a competitive, they can also, as described as competitive binding of miRNA. So practically what they do in this work, they try to describe this functional role, and they found that when the knockdown of the DLX6 antisense 1 inhibited the proliferation and invasion of esophageal cancer cells by enhancing the endogenous function of mTOR. But here I reported some of the result of one paper, for example, where you can see that the comparison, we have the comparison in expression profile in the normal tissue and the tumor, and also you can see the comparison in the different stage of the tumor, and is highly expressed in the tumor and in the late stage of the tumor. And the same, what they do as approach, they also knock down the non-coding, and you can see in 2 different cell lines, EC109 and KC-E30, and they demonstrate that the knockdown results in less proliferation in 72 hours. So and also and also they check the expression of this level also. So already they check the expression in cells, and then obviously they knock down to see what is the effect on proliferation, migration. So this is important, can be considered as a biomarker also for this cancer. But also in gastric cancer, we reported 4 studies, and also in this case, we have the upregulation of the non-coding RNA, and also the approach for the functional studies, mainly the knockdown. So in this case, for example, I reported the one of the article of Fu et al., where here we can have the comparison in adjacent tissue and the gastric cancer, and you can see here you can see the highly expressed in tumor, and also in this case there is a differences in expression in the late stage of the tumor. Also, we have that the patient with the gastric cancer with the high expression of DLX6-AS1 exhibited shorter survival time than those with a lower expression. And also is reported the expression in the cell line. So we can say that definitely this long non-coding RNA is highly expressed in tumors. What they did here, silencing the non-coding inhibited also the gastric cancer cell migration and invasion, as you can see on the right, where they reported the scratch assay and also the Transwell assay, where they silence the non-coding, and then they can see that there is a huge differences and significant differences between the samples. So Kian et al., for example, this is another article that we reported. They reported that the DLX6-AS1 was overexpressed and as well. And also the cell line modulates the glucose metabolism and shall grow in the cancer by targeting the microRNA 4290. And as you can see here, the putative binding sequencing was confirmed by the luciferase assay, and also the expression is inversely correlated. And so we can say that also in gastric cancer, we have this upregulation of this non-coding RNA, and also we can see that also in this case some of the study reported the correlation, the negative correlation with the target microRNAs. Another cancer that we also reported is colorectal cancer, and in this case we reported two studies. One is from Zhang, and as you can see here, we can also they also compare the expression of, between normal healthy tissue and tumor tissue, and also they describe that the long non-coding RNA can promote the proliferation and migration of the colorectal cancer cells, and here is also shown in cancer. Also, the approach that they had they overexpress the DLX6 antisense 1 in ACT116 cells, where they described the proliferation. As you can see from the plot, you can see that there is the proliferation, increased number of invasion, increased invasion, increased migration, and also inhibition of cell apoptosis. When they instead knock down the DLX6 antisense 1, they instead they inhibited the proliferation, invasion, and migration. So, and this is so it's clear that the effect of these long non-coding RNA in this cancer. And also, they reported that this long non-coding RNA regulate the activity of PI3 kinase signaling in cell line. Here is shown the Western blot, where you can see that in the phosphorylate protein, there is an effect from this long non-coding RNA, and also you can see in the left, you can see the overexpression or in silencing the long non-coding RNA. Other paper that I can report here is also Kong. For example, Kong showed that in CRC patient at advanced stage or with the lymphatic metastasis also has the higher expression. In that case, they reported the correlation, negative correlation with the miR26A, and there is so as you can see here from the plot, and also we can they described the they also reported the same correlation with one of the targets of 26. That A, that is considered a tumor suppressor gene in this particular cancer. Other cancer that I can mention, for example, is hepatocellular carcinoma. We also reported the other study for ACC and also liver cancer. Also, is the non-coding is upregulated, and the approach is in this case is all for knockdown. And in this case, for example, one of the one in Lee et al. Paper, they also reported that is, as you can see at the top of the slide, that is upregulated in tumor and also is upregulated in cell line. And in this case, the expression was linked to the microRNA, the expression and the effect on the tumor in migration proliferation was linked to the microRNA to 425.5P. And you can see here the putative binding site that they demonstrate they can demonstrate by the luciferase activity in the cell line and using the wild type construct and mutated concept to validate the result. Another work is also Liu. That Liu instead report also in vivo study that the DLX6 antisense-1 triggered the invasion and migration. But in this case, they is the target is the microRNA 513. So this is mainly all the paper that we reported in our article for the hepatocellular carcinoma. Also, another cancer that this is the one that I'm studying now here at the University of Sussex is also pancreatic cancer. Pancreatic cancers, unfortunately, is very difficult in in diagnosis, because it's discovered late, and then that's why is sometimes when it's discovered it's just in a late stage, and unfortunately has a poor prognosis with the 5-year survival rate very low. And but also in this cancer, has been reported that the non-coding DLX6 antisense-1 is expressed at high levels, and also is shown that is the high expression was positively correlated with the large tumor size at advanced tumor node metastasis TNMS and stage lymph node metastasis. So and the study that I here, for example, is also is demonstrated that is high expressing tumor And they, in this case, the function of this non-coding RNA is knock down the microRNA 81B, and that is a downstream target of the non-coding. So, so this is another. Sorry, I just lost. Okay. And here is another article from Yang et al., where also they here reported the high expression in tumor in all the cell line that BXPC3, Kaplan-1, and also they, with the Kaplan-Meier curve, they demonstrate that the patient that have a high expression unfortunately has a very poor survival and poor outcome. And in this case, also they reported another microRNA that is affected by the these long non-coding RNA, that is MIR496-5P. So this is, as I told you, this is all the effect of these long non-coding RNA on digestive cancer, but also in lung cancer. We reported several studies where the expression is upregulated, and also the expression is upregulated and in the different stage of the disease. Here, also in this case, we have that the difference is the comparison between normal and tumor cells where is high expressed, and Huang et al. Show that DLX6 antisense 1 induce cell proliferation invasion, while inhibit apoptosis through the regulation of MIR144 and upregulation of PRR11 in non-small cell lung cancer. And here, as you can see, we have the positive correlation between the mRNA and the long non-coding expression levels, and also show here the binding site of the MIR144 that they validate through the dual luciferase assay. Still, another interesting paper that we reported about the lung cancer was the Zhang paper, where they demonstrate these non-coding as a potential diagnostic biomarkers, and because they saw that the expression was upregulated. As you can see here, also we have differences in the stage, so early stage and late stage. The comparison, we can see that is higher in late stage. And what they did that is interesting is the fact that they also compare the serum the serum in highly expressed samples. They compared the serum in the patient, and they found that it was a highly expressed. And here we can see that the relative expression level between in pre-operation, post-operation samples decrease dramatically, and this is significantly different. So other respiratory system cancer that we considered, and also the expression is upregulated, is nasopharyngeal cancer, laryngeal cancer, sorry. And also the approach for the functional study was knocked down and overexpressed. Several microRNA was, so the function was regulated by the several microRNA like MIR199 or MIR26A, and Other cancer that we also considered was endometrial cancer and also ovarian cancer and cervical cancer. In this case, also the functional role of these non-coding RNA was in proliferation, invasion, and apoptosis. And also in breast cancer, we reported some study where also the expression was upregulated, and also the result in this upregulation is in proliferation, invasion, migration, apoptosis. The same, the approach was knockdown and overexpression. And also we can say that there is also in breast cancer the effect also on the therapy and the use of the drug mediated by the target, the microRNA target, like miR199B, for example, where there is that regulating this microRNA, there is this induced the resistance, cisplatin resistance, and also lead to the tumorigenesis and lead to aggressive tumor. Other cancer where we described the upregulation of the non-coding RNA, this particular non-coding RNA is in bladder cancer, renal cell carcinoma, and also prostate cancer, and also has been detected in central nervous system, where in the was upregulated in samples, so in patient, but and also they, as in the previous work that I just showed, they also evaluate the expression of these non-coding RNA in the cell line. About the functional roles, so we can describe the tumor progression, invasion, metastasis, and apoptosis, also EMT, and also in regulating the microRNA 5 or 6 TRPE. Also, it was involved in cell cycle and glycosis. So, is a broadly range of function that have these long non-coding RNA as in several cancers. So other cancer also in osteosarcoma is upregulated, and thyroid cancer as well, and also the functional roles is in cell growth, autophagy, stemness, proliferation, and metastasis. So we can say that the DLX6 antisense 1 has different molecular mechanism of action, as well as different function in cancer. As involved also in cancer epigenetics. There are here in these pictures, as you can see in this slide, is reported all the cancer that and also the targets and the mechanism that is we describe So, for example, mainly is the miRNA sponge or RNA decoys, or and also they is involved the signaling that the important signaling in different cancer that lead to high the progression of disease and unfortunately the poor outcome. So interestingly, we can here we in this table we reported the DLX6 antisense 1 as a competing endogenous RNA for microRNA for the microRNA. As you can see here with the arrow, I I find, for example, that there are some microRNA in common across the country. For example, the regulation of miR26A in colorectal cancer is also reported in laryngeal cancer and renal cell carcinoma, as well as, for example, for the neuroblastoma or pancreatic cancer, prostate cancer is the target was the MIR 497.5P. So means there is something that is in common, by the way, across the cancer. And so in conclusion, we can say that long non-coding RNA DLX6 antisense-1 is an oncogene overexpressed in multiple malignancies. As I showed you briefly in the paper that we reported in our article, that mainly the approach was knockdown. And when they knock down, they showed that there is a decrease in cell proliferation, migration, and invasion. And also is importantly how the DLX6 antisense-1 acts as cRNA to bind to different microRNAs and inhibit their function. And and also the upregulation of DLX6 antisense-1 result in inhibition of the expression of different microRNA. So importantly, the expression that is also correlated with advanced clinical stages, tumor size, metastasis, and also chemotherapeutic effect and survival of patients in different cancer. And what is emerged and what we think that is relevant from this paper is the fact that this is can be used as a tumor marker for clinical prognosis and therapeutic strategy of various cancer, as we reported. So thank you for your attention. I hope that I've been clear in this. Thank you.
Thank you, Paola. Matt, are you here? Would you be happy to run the Q&A session, please?
Yes, I am here. Yeah, thank you, Paola. A wonderful talk and great explanation of your paper. Thanks again for sort of describing it in a sort of different medium, just to how it's published in ERM. Have we got any questions that at the moment? I don't see any in the chat at the moment. Obviously, if anyone would like to mention any, would be welcome to receive them. But if not, does anyone else have any questions at the moment? Nicola?
Hi. Well, I've got several actually, but I don't want to dominate things. So, what I was going to ask first, they fall into two categories. One's sort of like the control of the long coding RNAs, and the other is their function. And so based on their function, do you think it's are there any normal proliferating tissues that also express high levels of DLX6 antisense-1? I'm thinking in terms of the bone marrow and the gut that are proliferative.
Yeah, actually, it seems that also in the paper we say that about this tissue there is nothing, not any reverse of. Yeah, and so it might be interested to understand exactly.
Yeah, because in your pancreatic cancers, although it was higher in the tumor, there was a significant amount of overlap, wasn't there, with the normal expression where you had those scatter plots. Okay, the means were statistically significant, but there were quite a few normal tissues that had higher levels than the tumor levels.
Yeah, this is true because there is an outlier. Obviously, you can have these differences, but I don't know if in the paper they reported why they had the outlier, how they justify the outlier. Actually.
Oh, it wasn't a single. There was, I mean, there was like in the graph that you showed, there was a scatter plot. There was like the tumor was like that, and the normal was, you know, the means were different, but the overlap was very substantial, and it very different from other tissues. Say like I think, was it the lung where you showed a very clear discrimination between the normal and the tumor, and I just wondered why that was.
Because also the pancreatic cancer, sometimes the tissue is not, you know, is first of all, biopsy is very difficult to get. And sometimes you have a lot, you don't really have just the tumor. When you probably it's not really is heterogeneous tumor. So probably it's this comparison is more tough to have compared to other cancer that is a very distinguished cancer, also because we know that in pancreatic cancer we have a lot of you know,
a variety of different cells. Yeah, that's true.
So probably this may be one of the explanations that we can have in this case, and because I don't know. Yeah, it actually is a very good question, and is interested to understand, but also they report that there is a differences. Obviously, compared to the other cancers, not so
it's not so clear cut, is it?
No. Other cancers are very remarkable, also in the. But I don't remember if also the cancer cell line, when they showed in the cancer cell line, they have, for example, the other paper in the Young paper, they have that is the difference is more remarkable, and also the p value is higher in, for example, in the Young paper, and also they report that the level of the expression across the cell line is definitely high. So, and also is what I can see is that in all of the cell line, you have the higher expression of this long non-coding RNA. And based on my experience of PDAC, we can say that the cell line, the cell line, the pancreatic cell line are very different because if it depends that they have a feature of epithelial cell line, these are mesenchymal cell line originally. And it's that's why I think that this long non-coding RNA is promising for this reason, because it's expressed in all cancer, is highly expressed, and also the effect seems consistent in all cancer about the proliferation, because in all the study that we reported is down regulation is not they use a knockdown and also overexpress, and the effect is the same in all the cancer that we described. So I think that is a really promising long non-coding RNA to consider for the clinic approach.
Thank you.
Thank you.
Yeah, again, if anyone has any questions, please pop them in the chat. Nicola, would you like to?
Yes, I'll carry on with the other side of my question, which was, well, what causes the overexpression in the first place? Do the genes coding for these long non-coding RNAs, do they have promoters? Or are there mutations, and is it hereditary? That was the other thing. So more along the genetics and the control of the expression.
I don't know what is the link between the cancer for the expression, but I think I don't know now, but Definitely is a good question, because I think that that can be related also for the transcription factor that and also from probably also the enhance factor that also can be controlled, because I don't know now actually, but I have to check which is the main factor that regulates and which is in common in all the cancer, because obviously each cancer is different for the expression of and overexpression of the transcription factor. So it means that there is something that.
Sorry, I thought you'd finished. Beg your pardon.
No, please go ahead.
I just, do they have promoter sequences?
I don't know, because what I found and what we describe in the paper is that we found that is as a different transcripts, and also this because for example another question was which is predominant, which of these transcript is predominant in the different cancer cell line, is all are expressed, all the, because sometimes in the different, also in the cell line, not only in patients, you have that the isoforms are differently expressed, and some of so would be interesting to understanding if there is a common, so this is common in all cancer, so or there is a different cancer as a different isoform that are prevalent to the other. And about the promoter, I don't know if really there is a specific study for the promoter of this that is very interesting to understand exactly the binding between all the several cancer and understand how is regulated in the cancer, what is in common. Yes. Unfortunately, I don't know. I cannot really answer.
I mean, you can go. I suppose you can always go back further and further and further. You know, what then makes the promoter be switched on? You know, are there mutations and things? Yeah.
And also the methylation probably, and there are also these other factors that we must consider, because it's important to understand also the level of methylation of the promoter. Probably, yeah. Yes.
Thank you very much. Yeah, so I guess that sort of rounds it off, Isabella. We don't seem to have any more questions in the Q&A box.
Well, in that case, I'll wrap up if no one else has any questions. I really hope this Q&A has covered a broad range of questions and answered anything you might have had. I really hope you have enjoyed and found it useful to hear from Paola and learn more about the journal. And I'd really like to thank Paola for today, for her time and taking part. Thank you so much. There'll be more webinars in this series to register your interest for. So please check out the information on the journal's website. The next webinar will be on Thursday, 26th of May, 10 a.m. UK time. We are also keen to keep our editorial team as diverse as possible in terms of gender, ethnicity, background, etc. So please do reach out to me if you or any of your contacts are interested. I'll pop my email into the chat for everyone to see. Feel free to also send me your questions. If there's nothing else, I'd like to thank you today for joining. Thanks so much, everyone.
Okay, thank you so much.